Hello everyone, and welcome to today's presentation. I'm excited to share with you some insights on the puC19 vector. Today, we'll explore what the puC19 vector is, its significance in molecular biology, and how it is used in genetic research. We'll briefly cover its structure, applications, and why it's such a popular tool among scientists. Let's get started and dive into the world of genetic engineering with the puC19 vector. Let's start by talking about puC19 and what its name really means. The P stands for plasmid, which is a small, circular piece of DNA that exists independently within bacteria. These plasmids are really important tools in genetic engineering because they can carry foreign genes into bacteria. Next, UC refers to the University of California, the institution where this particular plasmid was developed. It's quite common for scientists to name their plasmids after the institutions that create or first describe them. Finally, the number 19 is just a numerical designation, indicating that this is the 19th plasmid in a series or collection. So, putting it all together, puC19 is a plasmid developed at the University of California, and it's been assigned the number 19 to identify it among others. This simple name actually carries a lot of information about its origin and purpose, which is foundational as we explore how these plasmids are used in genetic research. Let's take a closer look at the structure of puC19. This plasmid is about 2686 base pairs long, which makes it quite manageable for laboratory work. One of its key features is its origin of replication, which is derived from PMB1. This allows the plasmid to replicate efficiently within host cells. Moving on, there's a critical component called the multiple cloning site, or MCS. It's a short stretch of about 2.8 kilobases that contains numerous restriction enzyme sites. This is really important because it gives us a lot of flexibility when cloning different DNA fragments. More sites mean more options for cutting and inserting DNA. Next, the plasmid includes selectable markers to help us identify successful transformations. Specifically, it carries an ampicillin resistance gene, so bacteria that have taken up the plasmid can survive in the presence of the antibiotic. Additionally, it contains the lacZ gene from E. coli, which encodes the enzyme beta-galactosidase. This enzyme hydrolyzes lactose, and in lab applications, we often use this to screen for recombinant bacteria by blue-white screening. Finally, the vector has a well-designed restriction site region, a 54 base pair long multiple cloning site, often called a polylinker. It contains 13 different restriction enzyme sites, including popular ones like EcoR1, HindII, and BAMH1. This diversity of sites makes puC19 a versatile tool for cloning various DNA fragments. Overall, these features, origin of replication, multiple cloning sites, selectable markers, and multiple restriction sites, work together to make puC19 a powerful and flexible plasmid for genetic engineering and molecular cloning projects. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's talk about blue-white screening. First, the vector design is key. The puC19 plasmid contains the lacZ alpha gene, which encodes part of the enzyme beta-galactosidase. Within this gene, there's a multiple cloning site, or MCS, where foreign DNA can be inserted. When we insert DNA at the MCS, it disrupts the lacZ alpha gene, which is crucial for the screening process. Now, onto the screening principle. Bacteria that have an intact lacZ alpha gene can produce beta-galactosidase, the enzyme that breaks down certain substrates. If the gene is disrupted because of our inserted DNA, the bacteria can't make beta-galactosidase anymore. This difference is what helps us identify successful cloning events. Finally, we use specific agents like xGAL and IPTG after transformation. The bacteria are grown on plates containing ampicillin, xGAL, and IPTG. Ampicillin ensures only bacteria with the plasmid survive, acting as a selection step. IPTG induces the lac gene system, turning it on. xGAL is a substrate that turns blue when cleaved by beta-galactosidase. So bacteria with functional lacZ alpha appear blue, indicating no insert disruption, while those with disrupted lacZ alpha remain white, signaling successful insertion. This color change makes screening straightforward and visually intuitive. Now, let's look at the results on the plate. The blue colonies indicate bacteria that have the plasmid without the insert. Because the lacZ alpha gene is functional in these bacteria, they produce beta-galactosidase, which breaks down xgal to produce a blue color. So, blue colonies tell us that the plasmid is intact and doesn't contain our inserted fragment. On the other hand, the white colonies are a different story. These bacteria have recombinant plasmids, meaning the inserted DNA fragment has disrupted the lacZ alpha gene. As a result, they don't produce functional beta-galactosidase, so xgal isn't broken down and remains white. This brings us to the key point in selection. When we're trying to identify bacteria that carry our desired recombinant DNA, we focus on the white colonies. They are the ones most likely to contain the inserted fragment we're interested in. By selecting these white colonies, we're effectively isolating the bacteria that have taken up the recombinant plasmid, bringing us one step closer to our goal. This visual differentiation is a powerful tool in molecular cloning, helping us quickly identify the colonies that matter most. Now, let's take a look at some of the key applications of puC19 that make it such a versatile tool in molecular biology. First, gene cloning is a primary use. Because puC19 has a high copy number and multiple restriction sites, it's ideal for cloning and amplifying DNA fragments in E. coli. This means we can produce large amounts of our target DNA efficiently. Next, in recombinant DNA screening, puC19 simplifies the process of identifying successful clones. By using blue-white colony screening, we can quickly distinguish between recombinant and non-recombinant bacteria, saving us time and effort. When it comes to site-directed mutagenesis, puC19 is very handy because it allows us to introduce specific mutations at precise locations within the cloned DNA. This is essential for studying gene function or creating specific variants. For DNA sequencing, puC19 provides a stable and straightforward backbone, making the sequencing process more reliable and easier to interpret. Finally, in subcloning, puC19 serves as a general-purpose vector to transfer DNA fragments between different constructs or vectors, providing flexibility in experimental design. Overall, these applications highlight how puC19 supports various stages of genetic manipulation, reinforcing its role as a fundamental tool in molecular biology research.